This short lecture should be review for most of you, but I found that there are enough students that either have forgotten this or maybe never learned it in the first place, some of the concepts here. So it's really central to our understanding of evolutionary developmental biology. This is kind of where it all begins. We're going to go all the way down to the genome, right? DNA, represented here by this double-stranded helix. And we are going to define what a gene is. So in simplest terms, a gene is simply a region of DNA that makes a specific product. And the vast majority of genes make protein products, but there are some other genes, like ribosomal genes, that make RNA, and that's the final product that is used in the organism. Before we begin, I just want to make a note that what we're talking about here is a eukaryotic gene. So this applies to all eukaryotes, be they protozoa, fungi, animals, or plants, but it does not apply to the bacteria and archaea. They have genes that have similar features, but do not have introns and exons and don't have a complex regulation set of regulation structures like we're going to look here. So just be aware that the prokaryotic genes come in one long strand without any introns. Sometimes, in addition to that, there are multiple genes that are all strung together and are all translated as one big long transcript in the prokaryotes, and that's called an operon. But we don't care so much about prokaryotic genes for this class because there's really not much form, morphology, right? We don't really have a developmental process in prokaryotes, right? Development focuses on that very short window of time from fertilization to a fully formed organism. And really, the vast majority of that developmental period takes place in the first part of development. So the first trimester, if you will, if we're talking about a human pregnancy. Most of that morphological structure is developed in that very short, important window of time. And bacteria don't do that, right? They do have some morphological differences, structural differences between them, but it's a very different process, and so we will not be using bacteria for our Evo-Devo course as subjects of study. So let's look at eukaryotic genes. Again, it's a region of DNA. This is not meant to be to scale. I put these two little dashes here to represent that many of the regulatory elements that control the expression of a gene can be quite a ways upstream. They're not just right next to the beginning of a gene. So the beginning of all eukaryotic genes is a promoter. It's a relatively short sequence of DNA that's fairly conserved, so we can often recognize promoters between very, very distantly related organisms. You can think of the promoter as a section of the DNA that serves as a signal, a starting point for transcription. The gene itself is made up of actually 5 different structures that I have not listed 2 of them here, but there is often, almost always, a short piece of DNA prior to the start codon that is the upstream non-translated region. So it's transcribed, but never translated. Sorry, it's the untranslated region, or UTR is what it said. So we have a 3' and a 5' UTR. So we have a short region on either end of the gene that's transcribed but never gets translated. They can be important, however, for helping to move transcripts and to process transcripts. So they're not really important for the final product of the gene. However, they can be important for proper handling and processing. So let's add those to our list, actually. I'll put that here. So a 3' and 5' UTR, or untranslated region. Now, sorry, let me finish this up. The 3' and the 5' refer, of course, to the side of the gene. So transcription can only happen in one direction. And so it's from the 3' to 5' direction. sorry, 5' to 3' direction is the direction that transcription occurs, and so translation occurs in the same direction. So I don't care that you know which end is which, but just know there's an untranslated region on either end of the gene. Exons are the portion of the final transcript that will be spliced together to make the final mature transcript and eventually be translated into the amino acids, the individual components of the protein, right? So exons are the coding region. Introns are non-coding regions that break up the exons. Okay, and this is not to scale. Typically, the introns are much, much larger, as many as 8 to 9 to 10 times larger on average than the exons. And there can be many of them. I've just, for this diagram purposes, put 2 introns, 3 exons, but there can be many, many more, or just a few. Nearly all eukaryotic genes have introns in them. It's a rather unusual way to organize an instruction manual, which is what the DNA is, right? But it is non-useful if you want to think about it. It does actually have a use, so that may not be the best term, but non-translated region of the gene that is spliced out before the mature mRNA is created. Okay, so we've talked about promoters, exons, and introns. And for most people, that is what the gene is composed of. The promoter, untranslated regions, and the introns and exons, that makes up the gene. All right, so just be aware of that. That's the strict definition of a gene. However, there are other important parts of the DNA that play a role in how that gene functions. So although these are not strictly part of the gene, we are going to include them here in our discussion of genes, and you need to be aware of their names and just in very general terms how they work. So the first elements that we are going to be talking about collectively are called cis-regulatory elements. Let's do those first. So cis-regulatory elements are regions of DNA. They're usually short, 6 to 12 base pairs long, sometimes a little longer, but a short region of DNA that serves as a binding site for a transcription factor. And there are 2 types of regulatory elements. So we'll talk about those very briefly in a bit, but let's just talk about the regulatory elements themselves. They are short. The term cis refers to, it's a term that means nearby or near to. So they are near to the gene. They can be a couple thousand base pairs upstream, but usually not much more than that. Occasionally, they can even occur in introns or downstream, but most commonly, the most of them are upstream of the gene, so somewhere near that promoter. And there are 2 types of regulatory elements. There are regulatory elements that bind activators, and they are known as enhancers. So together, the transcription factors that are known as activators and the regulatory elements known as enhancers, they work together to turn on gene expression. And so gene expression is just simply shorthand for the entire process of transcription. We'll look at that, an animation of that at the molecular level, but we're not going to worry so much about that for this class, all of the mechanics. I don't care that you memorize exactly all of the things that go into transcription. That's a process maybe more for molecular biology that you might learn in more detail there. But for this purposes, it's the big picture, the concepts that are important to us. Okay, so a transcription factor that is called an activator binds to a regulatory element that's called an enhancer, and the gene is turned on. Transcription begins, the gene transcript is made, and that will continue as long as that process is suitable. Now, in very simple terms, the other regulatory elements are called silencers. They also bind proteins called transcription factors, but a transcription factor that binds a silencer is known as a repressor because it turns off or turns down the expression of that gene. And there are often dozens and dozens of regulatory elements, some of them enhancers, some of them silencers, that all work together to regulate the expression of the gene. And so depending on how many activators you have bound and which ones, how many repressors you have bound and which ones, you will have a wide range of levels of the gene being expressed, being transcribed and made into an mRNA protein. Okay, so we've got all those vocabulary words down here. You should understand that expression refers to the transcription and eventual translation for most genes, although for some genes it's only transcription. And the process of regulation is basically turning on, regulating, right, turning up and turning down the amount of gene expression or the amount of transcription that is occurring. Now, this does also occur in bacteria, but it's a little simpler. There's not as many complex regulatory elements and different ways that genes can be turned on and off. Okay, so I'm going to put on a very short video here. I'd like you to just watch it. This is at the molecular level. And again, I don't expect you to memorize it, but I do want you to appreciate this process. It occurs very, very rapidly down at the molecular level. It really is a remarkable thing. So we are going to be looking at this process of going from DNA to the transcript or the RNA. I could put mRNA here because most genes, again, are messenger RNAs that are then translated into a protein. Some, however, do their final work as RNA and are never translated. So again, hopefully this is review for you. If you've forgotten it, review it. You do need to know it. So this process of making a copy of the DNA in an RNA version is called transcription. These are both nucleic acids, so it's really like transcribing it from one genetic language into the same or very, very similar genetic language. This is called translation because we're going from nucleic acids to amino acids. Proteins are very different from RNAs, and this translation process is a very different process than transcription. So take a look at this, watch this short video.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Okay, so life really is just a very complex series of chemical reactions, but by far the most complex chemical reactions that occur. And so you saw that was a very complex process of taking DNA and making an mRNA copy. Now that mRNA transcript will eventually be spliced together and then for most genes translated into the final protein product, which is the functional part of the gene. That's the gene product. That's what does the work. All right, sorry.
The central dogma.
Oops. So let's just talk a little bit about the genome, and in your next lecture you have some more information about genomes and transcriptomes and get some more familiar with those. But if we look at the human genome as an example of a eukaryotic genome, about 25% or so, I've got 26% here, so about a quarter is made up of introns. So those are classified as part of the gene, but remember they themselves never get translated, so they don't go into the final product, the protein product. This 1.5% are essentially the exons. All of the exons in the human genome only make up 1.5% of the total genome, right? So that's the protein coding region. But about a quarter of the genome is genes. So remember, introns are non-coding parts of genes. The rest of it's not really important for this class. It's important, and quite amazing that much of our genome is made up of non-coding DNA and structural DNA and other things. but you should know this 25%, a quarter or so, and then about 1.5% is the introns or the protein coding region of the genome.